point of my research that I've been working with, and I'm really interested in going on, is working with this Mito-Nuclear speciation. So mitochondria are very special organelles. I'm gonna go through them a little bit, and then I'm gonna show that using inspired models that are inspired by those ones that I showed yesterday. So I'm gonna show that I work with some a bit different framework. I've been studying how these interactions between mitochondria and nuclear DNAs can affect the evolution of species. So, so mitochondria, well, for biology has a primary importance because they are these organelles that you have like in several copies inside your cells, and they are responsible for the cellular respiration. So it's the production of energy in the cell. They're very small, they're very interesting because that's what provide the evolution of eukaryotic cells. So there's the whole theory of how what we call, like in the old language, superior forms of life, because you have like now big cells that have a nucleus and it can have like a whole machinery inside the cell. It was only possible because at some point the cells could absorb this thing that probably was a bacteria in the past, and once it was absorbed, now they can produce energy, so they can evolve to superior forms of life. Of course, this is not a very good language when they come, but well, mitochondria are very important for evolutionary biology. The thing is that in order to do this respiration process, in fact, it's not only the mitochondria itself that do all the process. You have some kind of genetic interaction. We have some genes that are activated in the mitochondria, some that are activated in the nucleus. Some proteins need like genes that are in the both nucleus and mitochondria to be expressed. One thing that's also very interesting is that the mitochondria have their own DNA. Why the nuclear DNA has like a very large quantity of genes, the mitochondrial DNA is very small, and it's very, very particular. It has very different properties. It's very small. It's a haploid. It has a high mutation rate, and so it's kind of interesting how these very two different DNAs, they interact somehow, and that's what provides the cell to produce energy. Given like the importance of this thing, the production of energy, also there's a whole study of mitochondrial DNA in evolutionary biology as to use as a marker of populations. So these properties of the mitochondrial DNA make it a good candidate to a marker of populations, in the sense that because it has this small size, several copies, high mutation rate, and is typically only inherited from the mother, you can trace the history of species, of populations using the mitochondrial DNA. For example, we can use the mitochondrial DNA to trace the origin of the Homo sapiens, for example. So this is kind of interesting. Not only that, but there is a particular...
I mean, naively, I would tend to think that rapid evolution is bad for tracing because you somehow lose track of who was your ancestors and so on, because there is rapid changes. So I would tend to think the other way around, no? I mean, suppose that passing from you to your offspring, it changed dramatically, become hard to see.
Oh, yeah, but...
I mean, in that sense, I would say that rapid evolution would be bad to trace back
The thing of rapid evolution being good is because since you have the alterations going on, in fact, there's the whole machinery of like how it makes the respiration continuous work. We have a selection going on all the time on the nuclear, the mitochondrial DNA. That's a thing that is part of the question. Like in the 70s, when you used to use the mitochondrial DNA as a barcode, in fact, there were some people defending that the fact that the evolution of mitochondrial DNA was neutral, because any like bad mutation would be fastly swapped out. But it's not totally true, because any kind of bad mutation could impair the respiration process. So rapid evolution in the sense, the mitochondrial DNA doesn't have our own machinery to clear these mutations that are bad. So they have to evolve together with the nuclear DNA. That's why it could make kind of a good, but there is not like is a gray area, like, because some you think that it works because it should be neutral, because the bad mutations would be swept out. But in fact, in more now modern ideas says no, they have to evolve together, and that's why it's good. It's a good marker.
And it's asexual. Is that what maternal inheritance means?
Sorry?
It's asexual. It does not require sexual. It's
yeah, the mitochondria they are asexual. Yeah, the replication of mitochondria is asexual. Okay, but per se we receive all the mitochondria from the mother, like. But it also varies a lot. Not cells in different kind of tissues have a different amount of mitochondria. So, for example, the liver is a the tissue in the liver has a lot of mitochondria because they need a lot of energy. Muscle cells, yes, sure. So that's what I was talking about. That since you have this whole thing about what happens to bad mutations in the mitochondrial DNA, because in the nuclear DNA, we know that there are some mechanisms of reparation, but in mitochondrial DNA, there aren't. So what happens, since you have a high copy, of course, when you have a bad mutation, you start some process that try to eliminate this DNA that had a bad mutation. But you can have effect in your cell. What happens is, since you have this interaction between the nucleus and the mitochondria, when you have some kind of mutation that make they being compatible, you have a decrease in the function of the cell. So you have what we call the Mito-Nuclear compatibility that's necessary, that affects the fitness of the cell. And this goes into several levels of organization. It goes like since from the cell, and if you think about the tissues, as I said, like you have tissues that are very critical, the energy production, and this goes, of course, affecting individuals, and then in a upper scale, it can affect the population. When as I'm gonna go through. So, for example, one thing that we can observe in this one kind of the animal, this is one model organism that we know that mitonuclear interactions are really important for them. It's the Tigropus, is an arthropoda. What you see is that if you have a population that have this compatibility and they are contiguous, in some point if they split, and the whole process of evolution, as I said, leads to two groups that still have a compatibility, but they are different between each other. And when they are put in contact, they will have these cases of incompatibilities. So they can run these experiments, in fact. If you have these populations, they still belong to the same species, but they have these different sets of mitochondrial DNA, and you do the crosses. what you see is in the second generation, you have offspring with a lot of incompatibilities, so these organisms are likely to not survive. So there is a hypothesis that, in fact, these incompatibilities have some role in the evolution of the reproductive isolation. That was thing that I was saying like yesterday. This is one known mechanism. So somehow when you produce these hybrids are not viable, so it's hypothesized that in fact these two populations at some point are going to become two different species. So this thing that occurs like in the cell level, somehow it may affect population level, it may affect the origin of species. So in the sense of all this, there is these questions. Why, in fact, the barcode work? So there was this idea that, in fact, the mitochondrial DNA was a good genetic marker because of this fast evolution, and in fact, no, maybe it's not. Maybe it's because it is interacting with the nuclear DNA, so it's affecting speciation, so maybe it's why barcode works. And by barcode, I mean that you have this use of using the mitochondrial DNA to identify species. This is something that we do. So we also want to know if you can identify signatures of this interaction, if this can somehow affect the species ecology, so ecological patterns of the population. And this can be summarized in the main question, like what's the role of those mito-nuclear interactions in speciation. So this is basically topic of my research. So for that, let's go back to the model. I'm just using a slightly different version instead of minus or plus 1, just 1 and 0, but the same idea. One thing that I didn't have time to mention yesterday, but mostly I work with finite genomes because they are here it's going to be important because I will have to write the interaction with mitochondrial DNA, as I'm gonna see here. But one thing is that we know from this model, from people before me who worked a lot with it, and we know that, as I said, like in putting all the rules of reproduction, evolution, and dynamics of the population, we can obtain speciation, and we can get patterns of species history, and these we can say, okay, some rules that we apply here are imprinting some patterns. So similar processes in nature should produce the same patterns. The idea is, even in a simple model, if you have like what are the important ingredients for the population dynamics, you should have typical signatures. So now I'm going to work with a version of a model that I have space. So now individuals are limited to a spatial region. So it's a square. Again, I have the fixed population of m individuals. As I said yesterday, it's individual-based model in the sense that I'm going to simulate individuals one per one. It's the biallelic genome, so each site is independent and it has 2 values, 0 or 1. And now the mating restrictions are not only genetic, I still keep that minimum similarity for mating, but also spatial. So anytime that one individual you reproduce, is gonna look for a mating partner inside the neighborhood. That's also one parameter of our model. So now I'm gonna also have sexes, so I have males and females. They have like finite DNAs, and similarly to what we had yesterday, when they produce offspring, it can inherit the gene from the father or the mother with equal chance, and has a mutation rate that gives the chance of mutating. Again, in the in a version of the model, you can eliminate fitness, say that all individuals have the equal chance of reproducing. But now I'm going to introduce the mitochondrial DNA. So it's typically a smaller chain. It has a mutation rate that's higher, and it's going to be directly inherited from the mother. So for each reproduction phase, I'm going to kill all the population, replace for the offspring, and I'm going to follow these rules to produce the genome of the individual. That's a nuclear DNA and mitochondrial DNA.
How do you distinguish between the two sexes? Is it one site along the DNA that determines if it's male or female? Or is it a particular, because they are painted in blue in the first thing, I didn't know if that meant that that was to identify, or if you have another tag.
I have another tag, yeah.
Okay.
I start with 50% chance, and then the sex of the offspring is also by 50% chance.
Okay, so it's a separate tag.
Yeah, but it's random. We keep, we don't have sex bias, so it's just by chance. Okay, so far? Okay.
Sorry, wouldn't it make more sense to have males have different genes, slightly different genes than females? I mean, at the moment, you can't distinguish a male from a female, right?
No, yeah. They are equivalent.
So none of the genes are correlated with any of the other genes?
Yeah, no. Which genes?
I mean, a male and female obviously have it's not just a name, a male and female, they have different genes.
They share 25 of the chromosomes.
Sorry?
They share 25 of the chromosomes.
Right, but okay. Anyways, I would have thought that anyways, okay, keep going. I understand the law.
Which genes, for example, would be different? Like less from the sex gene? Like
Yeah, the Y and the X.
yeah.
But the Y and the X affect other things, right?
Well, for us, what matters here is that the female is the one that's going to give the mitochondria. So.
Okay, go ahead.
Yeah, well, I'm not sure. Like if I can have a different sets when being a male or female would matters, but here I'm just interested in this property.
Okay.
But again, like a model, you can put all the elements you want to make it the most complicated as possible, but then it's difficult to separate things. I could put much more complications here, but I prefer starting to put the simplest thing.
Okay, thanks.
Okay. Then what I'm gonna do with this mitochondrial DNA, and then we're gonna now input some selection. So for each individual that has a nuclear DNA and mitochondrial DNA, we're gonna phenomenological formulate that mitonuclear compatibility by comparing these two DNAs and just checking if they have similar sites or not. Again, we are starting from the populations that are clonals. They start all from all zeros. So these ones, they appear only when they have some mutation occurs and pass through the generations. And so with time, we start to have incompatibilities. So we calculate this mitonuclear distance that counts from how many sites are different. And then we're gonna include fitness. So if before we had the individuals were equally likely to reproduce, now we're gonna have some normalization that individuals that have a higher mitonuclear distance, so they are very incompatible, they will have a lower chance of reproducing. So again, so how we do the dynamics? We pick one guy, we compare to the rest of the population, see if in the population how compatible it is. if he, by chance, still gets a chance to reproduce, he's going to look for a mating partner inside the neighborhood. And inside the neighborhood, the ones that have higher compatibility will have a higher chance to reproduce. And then we follow the rule of reproduction. Okay? So if we find...
For sure you said and got lost. Why is it important to have compatibility between the nuclear and the mitochondrial to reproduce? I mean, phenomenology...
we are using the principle that if you have these genes, this genome is too different from this one, the respiration process is more inefficient, so you affect the fitness.
I see, okay.
It's likely to not survive.
I see, okay.
I guess I didn't quite catch this earlier, but is that the actual mechanism in cells that makes this? Because you had the picture of the blue and the yellow cells in mitochondria, but is it actually true the nucleus of the cell has the same DNA in it as the mitochondria and they should be similar? Or is there some other mechanism of similarity that we either don't understand or it works differently?
It's not the same per se. It's a biochemical kind of compatibility. The proteins, they have to fit each other. Of course, as I said, this is a phenomenological approach, in the sense that it doesn't matter if even the nature of this gene is equal to this one. But I'm saying the protein that this gene is expressing is compatible to the protein that this one is expressing. And it is ultimately will lead to the individual have a lower fitness. Again, it's a much more simpler version because I'm showing one individual as one copy of nuclear DNA, mitochondrial DNA. So
you could think of these sequences as the protein encodings instead, and it would be similar.
Yeah, but what we are trying to say is this represents the survival of the offspring. If the offspring has more incompatibilities, it will be less likely to survive, to reach the reproduction phase. I don't know if I really answered. No, yeah, of course there are much more complications. We have several copies of mitochondrial DNA. You have a lot of other problems. For example, you can have cells that have different mitochondria, and you have a conflict. But again, I have to limit what is my problem here. So I'm assuming all the individuals is represented by this copy of DNA, this genome. And so I'm going to go to the results. We can come back to them all if you want to ask more things. But so as I showed yesterday, we check the evolution. Typically what we see, like here we have different curves for different levels of selection. So the red-orange curve, the higher curve here is no selection. And as we increase the selection, we see that the number of species that are reached are decreased. So this is what in evolution we call a radiation process. You started from a clonal population, a single species, and then with time, we see like this burst of species, and they reach an equilibrium, where the number of species are approximately constant. In this part of equilibrium, one analysis that we do is check how that's distance between different species. So remember, like when you have only considering that P of Q, that's the map of distance, but only considering different species. So when you don't have selection, we have that thing that the species, they are fixed. They are very distant from each other. So you have like species that are very distinct from each other. And what happens is when we introduce this selection, this is not true. Like species start to be more similar to each other. So it's much more difficult to species to break. because this interaction is somehow holding the evolution of the nuclear DNA, like saying barely. So that's why, since the mutations in the nuclear DNA are more difficultly fixed, because you have to keep the compatibility. So the individuals are more similar to each other. Speciation is hindered. So in the end, you have less species. Here.
Yes, sorry, I lost. Probably you said it, but what is physically this selection you're introducing? The selection that makes the interspecies narrower.
Here, when I do the reproduction, individuals that have higher compatibility, they have higher fitness. So there are two different things. Like when you do the reproduction, you compare the nuclear DNAs of two individuals, but also the ones that have higher compatibility, they are more fertile. They have more chance of producing offspring.
So and you can simulate this with
yes,
parameters?
Yes, purely simulation.
Okay. Sorry.
Yeah, there's the difficult because you have to deal with finite genomes. Because when inherent properties that mitochondrial DNA has to be smaller than nuclear DNA. This is important. So you can see that even if you consider not all the nuclear DNA are under this level of selection. You have some nuclear genes that are only considered for the species compatibility, but they are not considered for the compatibility with the mitochondrial DNA.
I have a stupid question. The blue, yellow, and red, what distinguishes them? I mean, one is 5 species, one is 15, one is 25. I didn't understand that.
Sorry?
You have number of species, and what is the blue, yellow, and red? Are these different simulations?
It's different levels of selection. So in this simulation, there is no selection. This has a weak selection.
I see, okay.
And this is a strong selection. Is that a parameter S? Sorry if I didn't put emphasis on that. But we control the strength of selection here.
Okay.
So if there's no selection, this term is 1. You recover the situation that everyone is equally likely to reproduce. As the higher this selection factor, this compatibility will count more for the fitness.
Okay. Can you just go to the next slide? So it seems like it all happens in like 2 generations, right? Everything is decided in 2 generations. It's like from the 10th,
2,000 generations.
No, but from the 10th to the 20th generation, everything is decided, right? That's where the difference between
here
the
I
3
think it's 100 generations.
Okay, so in 100 generations, everything is decided. It seems like it happens at something drastic happens that decides if it's going to be 5 species, 15 species, or 20 species.
In fact, I can show you later some other results. We see that where this evolution stops, like what we reach as equilibrium, in fact, is kind of independent what we have here. Once you have...
I'm just looking at the graph, right? Something special happens at about 100, it looks like maybe 50 generations, yeah.
Yeah, what I'm saying is that for this situation, when we don't have selection, it's much easier to produce the species to appear.
I understand that, but I'm asking why something special happens. It looks like everything is decided in the...
No, yeah, because remember that mutations have to... Here, like all the populations are the same, like all zeros.
Yeah.
What you make species appear is mutations that are accumulated and make the species break. If you have the selection, it's much harder for these mutations to accumulate.
I understand, but it's a little bit curious to me that everything happens then, and then nothing changes.
I think we're looking at exponential growth on a linear plot.
You mean like why this is so? Yeah, but it's a phase transition.
It's a phase transition? Is that what you want to say?
Yeah, well, I could use like enhance here and check how many generations it takes to reach this peak, but
Okay, keep going. It's surprising. Wait, keep going, please.
Happens in a very narrow.
Yeah. Well, okay. If I could like enhance this, I could see like you can see that. Okay, so here in the beginning you have some ephemeral species. Sometimes they start 2, then 3, then 2, then 3. Maybe the scale here doesn't show like, okay, maybe in fact, if you enhance here, you can see that there is a steep. It's not like from one generation to another. Maybe, but I know that maybe some previous work, this is not my focus, so I didn't see, but it's not like instantaneous, if you say that, okay? It's not from one generation to another.
And, I mean, that's a transient. You're not interested in the transient. You're interested in the asymptotic, say.
Huh?
No, that's a transient, no?
Yeah. It's not, yeah.
It's everything. The height of that peak is where it determines if it's going to be 5 or 10 or 20. It's not a transient. It's not something that goes away.
What is a transient?
Maybe I don't know what transient means.
Zero species is not an equilibrium.
Oh, okay, maybe I don't know what transient means. But I'm saying everything is decided in those 10 or 20 generations. That's all my observation. Okay, keep going. Sorry.
Yeah.
You write the I guess you go back one slide where you.
Oops.
No. You write that the fitness goes like e to the minus s d squared. There it is. Good. So naively, I might have expected d rather than d squared. That I would just count the number. So every incompatibility would make a contribution, and you add them up.
This is just
Whereas
because
this is
we
what I
wanted
guess
to make a Gaussian, like a
This is what would people in your field is maximal epistasis or something, right? Because the every pair contributes. Things go as the
okay, yeah,
the
it's too
pairs
severe.
Of
Yeah,
it seems very, and I don't know whether it matters. So I was wondering if you put D there, would it make much difference?
Yeah, it's just because I wanted to be. I only think about the Gaussian fitness distribution because I wanted to have like not only decay. I wanted to have some. I thought this selection as a width, but I never thought about that, to be sincere. Like for me, it was always so. Okay. Remember that it's still like it's not. Still, some individuals have chance of reproducing. Like they're not zero. Right. They have a reduced chance, but they still reproduce even if they have a.
So I should think of it more as what 1 over the square root of s is my tolerance.
Yes.
For
yes, that's it. Yeah.
Okay.
So that's what we were talking about, the radiation. That's what in ecology we call radiation, when you have this burst of species. Surprisingly, we see this in nature. I know this is something that maybe is weird, but this burst of species, they are documented. Like so, in fact, it's very useful for us to compare to real systems, because this is how we get like those nice phylogenetic trees, and when you can compare the topology of real trees. But just for the sake of the simulations, analyze what's the typical topologies that we obtain from these simulations. So we use these two kind of metrics to study our systems. That's the balance of the trees, and how long are the branches? If they are this is very long, so they are TP. And then you can see that the phylogenies that each system produces, they are quite different. So we have like this distribution of, when you don't have selections, you have trees that are basically stemmy. They are either balanced or unbalanced. But when you have selection, they are very tippy, indicating that the species, they are typically short-lived. Okay. So, and then we also analyze what is this equilibrium. So we see that the number of species remains in the average value, but there is oscillations. What's going on in this equilibrium? That species are still appearing, and some are being extincted. So it's a dynamical state. When species are appearing, some are disappearing. Also, we count here for situations, and we have the split of species, but after 2 to 10 generations, sometimes they join together. So this is also something that happens here. Like this process of speciation, it's not instantaneous per se. Like we have some species that split, and then some mutations can occur and bring them together. We call this hybridization. So we can count the rates in each situation that species are created, so species are extincted, and the species are hybridized. And then we see that when we increase selection, all these rates increase. So in fact, here, it's really like a very soft situation. Species are created in a very low rate. They basically have a very low extinction rate. They are very permanent. But once you increase selection, you increase this exchange of species. They are all the time being extincted and being replaced by new species. And the hybridization also is more likely to occur. And this is what we call the macroevolutionary turnover, this exchange of species. And this reflects in the lifespan of the species. Like the ones that are here, they have a very long lifetime. But in the lifetime, I mean by the moment they appeared, and they still produce the offspring that are from the same species. But these ones that are under strong selection, they are very short-lived.
Sorry, Ivan.
Sure.
So you have geographical distribution also, right?
Yes.
This is spatial also.
Yeah.
So you have different species in different corners of your...
Mm-hmm.
And the question is, how do you distinguish different species?
From the criteria of genetic distance.
Yeah, but there has to be...
Yeah, we compare all individuals. We compare all individuals. So imagine that you have individual 1, 2, 3, 4. We compare them too. If they are compatible, they belong to the same species. Then we compare this individual with this, with this. If it's compatible with any of them, it belongs to the same species.
So a species, so there has to be a boundary, right? Something is, and at the boundary, one is very close to another, right?
Oh, yeah, I don't have the figure here. Yeah, when you have the simulation, you can see that they are localized in space, yeah.
But the question is, I guess my question is slightly different. There should be a continuum of colors, right? Because you're trying, you have to say like, okay, from 1 to 10 is one species, from 11 to 20 is another species, right? Let's say instead of being binary, you did just numbers, right? Just to simplify. Suppose for a binary, you know, the 2 to the, there's a way to get a number out of a binary, right?
Yeah, but they're doing edit distance.
I know, but...
Yeah, it's comparing pairs of
Right,
individuals.
right. So the question is, aren't there things which are very close to something of another species? By definition, it can't be, right? I'm not sure I'm asking my question in a clear way.
But it's like this. When we map the genetic space, we compare the pairs of individuals. So basically, it's that matrix of similarities, and we group together. We make like clusters of individuals to have like the possibility of gene flow. Sure.
Okay, so I know from the talk yesterday that you defined the species by a cutoff in the overlap, but it's not unique, right? 2 species can be close enough in overlap to reproduce, but with with a third, but from each other.
So yeah, so that's why I put here this network. You see, like, for example, this individual cannot reproduce with this one.
I see, okay.
But you have,
So,
like, the possibility of gene flow.
So you call everything that is connected in the graph of things with that exceed that overlap to be the species, even if not everyone can.
Yes.
And do you let the ones
like
that can't reproduce, or do you only, the reproduction rule doesn't change?
Sorry?
So for the sake of counting species, you do this.
Yes.
But for the reproduction rule, you still use the Q cutoff.
Yeah, yes. Like when I do like the reproduction phase, if I pick this one and this one, they won't reproduce. But when I classify species, I count as belong to the same group because you still have individuals connecting them.
Okay. I actually have another question here, sorry, which is, in these simulations, the data you're showing us, what is the ratio of lengths to the nuclear and mitochondrial DNA?
I use 1,500 sites for the nuclear DNA, for the mitochondrial DNA, 500 sites.
So three to one.
Well, yeah.
And is that close to your model organism?
No.
I guess, so my question is do these results, do they persist to infinitesimal lengths of the, because it seems like the mitochondria is much shorter. So if you take the limit of mitochondria being much shorter than nuclear, does any of this survive? Yeah,
the problem is when I start to work with this, if you make this too short, you start to have what's the name? We start to lose information because once you have a high mitochondria, the mutation rate is too high, there is a upper limit, a lower limit for the mitochondrial DNA in order to not lose the information. I don't remember what is this name, but it was in the complexity book of, well, I don't remember the name. But yeah, I made this study and I fixed the length of the mitochondrial DNA. And yes, of course, I can make the nuclear DNA much longer. I had some examples, but the thing is the phenomenology is hold. So the thing is, like, I can change the mutation rates, sometimes the length of this change, but the phenomenology holds. So if I make like a much longer DNA, the problem is that all these phenomena will take longer to happen. We know, like, we studied this problem for a long time to know how it scales. We know that it scales, like, that's a thing. Well, another thing that you have to think, the nuclear DNA is much longer, but not here consider that this is only the sites that are important for reproduction. So it's not like the nuclear DNA, the whole nuclear DNA. It's the ones that are being selected for reproduction, kind of.
Sure, I think you told us in the first slide, like 30,000 genes versus 70 genes
Yeah,
or something,
yes,
right?
20,000 to 37.
Okay, so
yeah. Yeah, in fact, when you look into the genes that are really interacting between mitochondria and nuclear DNA, they're not so different from this proportion. Because remember, you are considering here that you have like 500 genes from the nuclear DNA that are interacting and another 100 that are not. And I could put much more that are not. But basically, what I want to see is the results of interaction again. Then I will have just increased the selection to see the effects.
But this is the important point, right? It's possible, I don't know because I haven't seen the simulation, but it's possible that in the limit of having much more non-interacting DNA, that your effect shrinks to zero with the ratio, right? And the question I was asking is it true that in that limit of the length of the non-interacting DNA becoming much larger than the length of the interacting DNA, does your effect survive?
Oh, that's interesting.
Because that seems like the relevant limit to actual life forms that have this property, right?
Well, you said something about the things taking long.
Yeah.
The effect goes down with the possibility of taking longer to study it.
I don't know.
And also have to consider that when you think like, okay, I'm going to have a size of DNA, this is a problem per se, because size of DNA is not something homogeneous in nature. You have yeah. So yeah, for the model organism, this thing kind of fits in the sense that we see the proportion of the sites that are interacting. But my guess is like this would be a problem of scale. I think that even if I put a lot more of sites here, much longer DNA, then I will have to just adjust the strength of selection. I don't think they will go to 0. Even because, as he pointed out, that selection that I'm putting this distance is quite strong.
So I can ask another simple question. So let's say you need 10 genes to be the same in order to be able to reproduce. So the yellow and blue, let's say the closest the yellow and blue could have is 9 genes, right, in common. But the yellow with another yellow that it can't reproduce with may have only 5 genes in common, right? So it's not clear to me that the yellow with yellow has more genes in common than the yellow with blue. There might be some yellow with yellow that have less genes in common than a yellow with a blue.
Oh, you can have overlaps. No, then no. If you have one individual here that has like the similarity below the reproduction threshold, it's not possible. We don't have.
Right, I understand that. So let's say it needs to have 10 in common in order to reproduce. So let's say one of the yellows has 9 in common with one of the blue, it can't reproduce. I understand that. But if you take another of the yellow that it can't reproduce with.
Okay. This one and this one, for example, 10.
Yeah,
but
they might have less genes in common than the yellow with blue, but you're calling them both yellow.
But yeah, because with this group, he is not similar to any of them.
I understand.
So you seem like you think that he should be.
There should be some intermediate thing. But
yeah,
in any
it
case,
should be isolated.
Well, no,
again,
it could be green.
Yeah, well.
I'm just saying that your definition of
this
species.
That's a thing that how we define species.
Yeah.
We chose a definition of species based on this this genetic network.
I understand.
Yeah, but it's a choice.
Okay, I understand. I just want.
We could be more soft or even more rigid. Like once this guy cannot reproduce, we have only like fully connected networks of individuals.
Okay, thanks. I just wanted to make that point. Thanks.
I have there's a whole paper, like only of definitions of species. I was just talking yesterday to Will, like in nature, species are like a weird thing, because sometimes you pick up individuals that are super different, you put them together and they reproduce. So we have to choose one concept and apply for the model. And this is like, with this, we cover a lot. That's what matters. Then, comparing that thing with no selection and strong selection, then we have the model organism, and we have, this is the typical distribution of subspecies of the model organism. And we did the test of having the similar spatial distribution and run this with no selection and strong selection. And this is what we obtain with the strong selection. We adjust the model to have like the a similar number of species. And what we saw is that when you have the selection, there's a higher correlation between the position of the species and how they are related to each other. So if species are like spatially distant from each other, their common ancestor is much older. If they are close to each other, it's likely they are more recently related. And this is something when you run without the selection, this kind of loses. So this, when you run without the selection, this kind of become all entangled. You have sometimes species that are super distant from each other on time, but they are neighbors. So what you saw is like mito-nuclear selection hinders the speciation, makes the speciation harder, but during the process, it creates a higher correlation between the species history and their relation and the spatial location. So that's, we suggest that this is maybe a cause why the barcode works, because it's creating a correlation with space. Another thing that we wanted to understand is this thing that's kind of non-intuitive. You have a high number of species, but the speciation rates are low. And this I put like as bold insights because it's really something that we are suggesting. Let me explain. We have these patterns of in ecology and in evolution in the world that call global patterns. One important global pattern is, as I explained, I mentioned yesterday, is that near the equator, typically we have more species than in the poles. And these have several explanations. Like Nathan mentioned, we have the same models, we have the same results. Well, in biology, it's kind of that. We can have like several explanations why this happens. Really, like there's a whole discussion. You can understand like, oh, these regions of the planet are older, these regions of the planet have more resources, so they have less competition. These, the behavior species in the poles are different. You have a list of reasons that why that could happen. And not only that, but this is not patterns that are not homogeneous. Some species, they go against the patterns. For example, corals, they don't follow this pattern. So that's why models are important to help us explain what are the main processes that are underlying the patterns that we observe. So this is a kind of a bold insight that we had, that we know that these species, that we have like a lot of species in the equator, but weird thing is that in this region, typically we have lower speciation rates. And in fact, these regions that are that has lower richness, they have higher speciation rates. Also, there is some ecological reasons, ecological explanations for that. But one thing we suggested that maybe here in these regions, because you have a scarcity of resources, this selection for mito-nuclear compatibility is more important there. Because you need energy. Like if you are a bird, for example, you have to fly long distance. Your mito-nuclear compatibility is really important for birds, for example, because they need to be very efficient in the energy production. So if you could think that, in fact, here you have a region that has a lower selection because you have like a plentiful of resources, and here higher selection, you see a connection between a metabolic mechanism, that's the energy production, and a global pattern, that is how it's affecting species in a macro evolutionary scale. And also, this is the same for ages. We see that the species that are typically in the temperate regions, they are younger, and the species that are in the tropical regions, they are older. Again, this is the kind of thing that you have several explanations. What we are saying is our model proposes a connection between things that happens in in the molecular level, in the genetic level, and the scales to species. Sure.
What is going on with sub-Saharan Africa here? It's completely, it sometimes is following what you're saying and sometimes not following what you're saying on this plot.
Yeah,
It has,
as I said, the patterns are not homogeneous. Like these were made for birds, if I
so
remember.
sub-Saharan Africa has horrible species richness for birds.
Yeah, that's why also they call, one of the theories is that the species came from here, they migrate to here. So that's why they kind of are not following the pattern. Yeah, I wanted to focus on this region that I put the
Sure.
the arrow.
Africa is, I'm wondering if there is a data limitation because it's not as well mapped as other regions of the world. I don't know.
I think, well, of course you have some limitation of the data, but that's why when you're very careful when you say, like, you're seeing this pattern, and then you limit, okay, for this phylum, for this group of individuals. For example, as I said, corals are are kind of individuals that don't follow this pattern. But
I'm just wondering about the
I think that
the
in
data
this case,
collection, if the data is collected equally, I mean,
in this case, is because they chose birds to make these patterns, and the birds that were related to each other, like this was based like in a whole, in a single phylogeny, for example, like in terms of relatedness between in a single phylogeny. So this saw the birds, like for example, that live here are not related to these ones. We chose one phylogeny to do this study. But yeah, well, in fact, this is a problem in biology, like because we cannot run the experiments in the lab. We count from the fossil record, and there's a whole discussion, even how you obtain for example, species ages. This is not something. We go there, we clearly see we have the whole problem of inference. This is something that also we mentioned, like there is a paper about how, if you choose a different method of inference, you can have different phylogenies produced that would have the same data resulting. It's like different process will produce the same data. So that's the thing that's why models matter in biology, because we have to discuss things. Like it's not a closed answer. It's not something that we have a closed answer. It's not an exact science. But yeah, you have to count like for, if maybe you don't have enough data.
yeah.
But I don't know, maybe somebody tomorrow discovers a bird here that ruins completely this thing. This may happen. Yeah, but yeah, there's a whole also discussion about how in the global south, for example, you have like yeah, well, I don't know if I have much more time. Well, I had this thing, but I think I'm gonna. I want to just have more time to think, but just to connect with what we talked yesterday. I think about what I still want to study in this system. One thing I can say is that there are not much people studying the influence of mito-nuclear interactions and speciation in the modeling part. Like there's a lot of people doing lab work, but there is a scarcity of models. So when you say to me, like, I come here to present and I have some new insight of something that I have to study, I think that I still have a lot of room to work with this model. And some ideas they still wanted to explore. As I said, like I can have, for example, a much more soft definition of species and see if it is changes everything. Like one thing that I wanted to do, for example, is making the selection much more soft, not hard, or even make it like inhomogeneous space and see what happens in the frontiers. Because also we know that mito-nuclear interactions are really strongly connected to hybridization zones. As I said, like I saw that increases the probability, the rates of hybridization. So we have the whole thing of studying how is the genetic patterns of hybrids in these frontiers. Also, I said like I do the selection over the mito-nuclear compatibility, but there is other levels of selection going on. Remember that the respiration depends on the combination of them, but also part of the respiration is only done by mitochondria. So you have some selection for optimal mitochondrial genotype. So one thing that I also wanted to consider is consider that you have like optimal mitotypes and see how this affects. And so including another level of selection. And connecting with the islands, now that I have this one form of selection in my model, I wanted to see how this affects the colonization, like, and even different sets of islands. Like we have the 2 islands model, we have this the linear or the kind of circle, or this kind of system that in fact is quite important. We have a lot of observations on this kind of thing we call stepstone, that when you have a bigger island doing sequential colonizations of smaller islands. And this is like, it has a lot of potential to compare to real systems. And I think I'm going to stop here today. Okay. I don't know, you have more questions?
So we have time for questions. Yeah, go ahead.
So actually, two questions. One is, yesterday you mentioned that there was some work where they had some model and they managed to predict species all across the world, but you said that model had too many parameters, and
yeah.
maybe you could comment a little bit on what is that and how does it compare with what you do, and does it have too many parameters, but is it a good model in the sense that it does describe everything very well, and therefore from that point of view it's done, or it has flaws and this is better and so on. But and then the other question I would have is, are there corners of this quantitative ecology business that smell more quantitative, like with power laws or phase transitions or something, that would be a bit more robust that one could try to track rather than curves that have time scales?
Yeah, thank you for your question. The thing is what we aim with the model. Maybe, sorry if I want to look like I want to diminish the other model. No, the guys are really good. They have it's called Genesis. It's kind of interesting model. But it's the purpose of the model. Like, for example, those guys who made the model, they are more interested in making predictions, for example, under climate change. So once you have a simulation of the world, you can just check what's going on when you have some change of temperature, like circulation of oceanic waters. What is the purpose of their model? So, but I mean, the downside is if you have a model with too many parameters, it's hard to separate what is important or not, what you want to learn from the model. So my model, I want to learn about the process, guiding, and what kind of patterns I have in result. So I study speciation per se. I want to understand what are the important things that guide speciation and what kind of signatures I have. So I think there are different purposes. One thing is when you have a model that's too big, is of course difficult to run. It's a massive time to simulate, it's computational power. And, but yeah, for me, I think it's like different purpose. But I have someone who I work with that you can have like comparisons of the models. Once you have that model that is super powerful and run and you can have all the control, you have a simple model and then you can do this filtering of the conditions. So you can compare them too, like, or using one as input of the other. But yeah, and the second question, what it was? More quantitative, yeah. In fact, this is some, I work with this model since 2018. And this is something that once I become an independent researcher and I have like independence, I would like to do, like something more robust, something that I can do quantitative approach, like, because this is very qualitative. And it's not bad, like in ecology, in biology, a lot of things are verbally explained. Because, for example, one thing that I want to study is if the symbiosis between nuclear DNA and mitochondria could have some impact in the evolution of sex. This is something that you don't have data. Like you have some organisms that is scarce. You cannot run an experiment. So if you can do something that even gives you a qualitative insight, can be interesting. But yeah, in the other hand, but I think that making something more robust and quantitative is something that will take time, and I think I will need like to have a structure for myself to do that. But it's something that I want to do. But I have to admit, I don't know exactly how, but I want to.
Maybe a little bit related. So you were showing us the west coast of the United States,
Yeah.
and the relationship to your predictions. So maybe have that there so I can think about it. Okay, so.
Yeah, it's the continent.
Yeah, no,
the one
before.
with, this is California, yeah.
Right. And so, I mean, the whole world seems maybe a little overambitious because things aren't uniformly accessible, whereas I might be willing to believe that the migration of species, since the mountains run parallel to the coast, right, I could believe that you could go this way more or less uniformly, whereas going this way is complicated. So, but I worry that sort of the environmental difference between, I was going to say San Diego, but you actually go all the way down to Baja and Seattle. Those are very different environments.
Yeah.
So how is that mixing with your ideas about speciation? I mean, if I forget about mitochondria and I forget about selection and everything, I could just say, well, look, it's cold in Washington, and it's rainy, and it's hot and dry in Baja, so I should have different kinds of plants. And is that just a different axis, or?
Yeah, what I'm saying with my model is that I don't know, like, if there is an optimal mitochondria type, but for sure, they have to be compatible. But see that even if like they're very different, they still belong to the same species, and they have this hybrid breakdown thing. Like when you mix these guys with these guys, what you see is the problem of the hybrid incompatibility. So I'm assuming that if this was the problem, this was the thing that's going on here. That's the thing. But yeah, you could say, like, for example, here they under some selection that's much stronger than here, maybe.
And do you know anything about the evolutionary tree is built from what part of the genome? Is that built from the whole genome? It's built from, or it's?
These ones. Let me remember, because I got the contact of the guy that built this thing.
I guess what I'm
I
wondering
think
is...
it was the whole genome, yeah.
Okay, so I guess what I'm wondering is, if you're saying that mitochondrial nuclear interactions are an essential component of speciation, it would seem like what you want to do is to take a situation like this, where you have species spread out geographically, and sequence the mitochondrial genomes and see whether, I mean, I guess you have a prediction for how they should, you know.
Yeah, but that's what people do. They build trees based on mitochondrial distance. Like, they build trees using the mitochondrial DNA.
So do you have a prediction for, let's say, relatedness in the nuclear genome versus relatedness in the mitochondrial genome?
Yeah, and that's also a thing that there's one guy who works especially with these Mito-Nuclear things. He's an ecologist. He works with birds. And he wants to propose one type of species that was the Mito-Nuclear species, that in fact the species would be the sets of individuals that have this compatibility, for example. So yeah, again, this is not uniform. For example, for plants, they don't care at all about this Mito-Nuclear stuff, because they have a whole different process of producing energy with photosynthesis, and you have a third set of genomes that is from the chloroplasts. But you have for some phylos, using the mitochondrial DNA is more reliable to build the phylogeny than in another phylos. Yeah, but that's one of the questions. Like in the 70s, when Avis was using this as a genetic marker, this was a question. Like, this is how reliable is this? Like, how, because of this thing of compatibility. And you know that for some living beings, it doesn't. You can have problems. So, but for this one, now I think this tree was built based on the mitochondrial DNA. But yeah, mostly evolutionary biology used the mitochondrial DNA to build the phylogenies.
A very naive question. When you showed the plot about the growth of the species in your, is there any explanation for the fact that you have a huge spike and then the drop that you see after the initial growth is?
Yeah. There was a previous work from the group, without including this thing, when this has there's a name in ecology, in fact, that we have a peak, and they study all the conditions when you see that this peak is larger or not. Like, sometimes it's affected by the range of the mating neighborhood. Yeah, there's a whole thing. But in my case, I was interested in the equilibrium.
Okay.
So, but for example, and I would say that why I am interested in the equilibrium, because I have this work when I started from populations that have some mismatches in the genes. Instead of starting, and I wanted to see how it affects speciation. And one thing that I saw is that when I have the mismatch, when I have higher selection, in fact, the speciation occurs faster.
I see.
But the thing is, the equilibrium is unaffected.
I see.
So the equilibrium is determined by the parameters of the system. It changes a bit what happens in the beginning, but the equilibrium is the same. But yeah, so this is basically determined by your initial conditions, I would say.
Yeah, I see. So
you see
essentially
that you have like these different shapes here.
Your understanding is mostly late time, so once you arrive at it. Okay.
So this model you're describing has a few parameters, the radius, the ratio of lengths, the Q-min, all these things. How do you select these to compare with something like this model species? So you made a comparison with a model species.
Oh, yeah.
How did you do parameter selection for the simulations that you were comparing with that? Because you have the R and the cumin and the.
They are qualitative. That's the. We run several simulations. As I said, like to get, for example, this case, we had some simulations like for with this stripe, and we kind of know the conditions that give like what the number of species that we can obtain in this distribution with this structure. But what I can say from robustness is that I know that when I make variations from that, what I get is a comparative of the things. For example, if I changed everything drastically, I still would know that. If everything changed drastically, I still know that the fact of selection is decreased. Maybe the number of species is different, maybe it's taking longer to obtain. But in the end, what I say is, when I increase selection, the number of species decrease. When I increase selection, these things hold. It's a comparative study.
Fine. And this is just a curiosity. So is it true that like, have people done studies among like people? Do elite athletes have this mitochondrial nuclear matching? Is this something that actually matters for like human athletics?
I think it's more about the quantity of mitochondria.
Whether they match or not seems less important. I don't know. That's been studied.
Well, there's a lot of mechanisms of inside the cell to compare. One thing is that studying this compatibility for human diseases. This is something that people are really interested, because mitochondria is also associated with aging. Because since you don't have the repair mechanism, when they accumulate a lot of mutations, mitochondria, they kill themselves, they kill the cells, and you have problems, for example, heteroplasmia. Like you have different copies of mitochondria inside the cell. This can have some disease related. One thing we call mother's curse, because since you receive your mitochondria from the mother, there's interaction with the X chromosome. So the Y chromosome, sometimes there are some genes that has epistatic bad interactions with the mitochondria. So men are more likely to have mitochondrial diseases. So it's quite a tense thing, like a topic. And maybe in what sense you were talking about the difference between males and females. For sure, one of these things, like males are more likely to accumulate deleterious mutations in the mitochondrial DNA.
So you chose this problem, but the question is, I mean, if you're a physicist, you try to choose a problem in which you can test. It sounds difficult. I mean, is there any lab in which you could, I don't even know, does evolution appear at a lab scale? Or are there no things that you can actually speciate in a lab?
Speciate in the lab, you were talking yesterday about bacteria.
So do bacteria speciate? I don't know.
I'm not, because I'm mostly.
That I believe.
when I try to look for model organisms, I try to look for animals. I know like the cases, like, but when you see like few generations, it's not in the lab. Like mostly when you see like speciation going through few generations, if someone goes in the nature and observe it. I know that, for example, in Galapagos Islands, they know that some birds speciate in few generations, and lizards, I think, also is easy to, they speciate in few generations, but it's not something that you do in lab.
What about plants? Can plants speciate? I have no idea. I mean, I'm completely ignorant, but it seems like it's very difficult to test the theory.
Because we don't have labs that can run millions of years. So we have the whole problem that we have to deal with the data that you already have.
Yeah, but No, but
Yeah, we have to take a fossil.
No.
And the problem is like
Cosmologists-
fossils.
No, cosmologists have telescopes, right? You don't. If,
You can't like tweak the universe.
no, that's not what I'm asking. But I'm just. I mean, you chose the problem. Is this something you're going to be working on in 10 years, or, I mean, or maybe your supervisor chose the problem? I mean, it sounds like a problem that, if I was a physicist, I would say, well, this is a dead end because you're not going to be able to test it. I mean, so you can.
It depends on which kind of curiosity we have. Maybe I'm more like in a biology now than in physics, because biologists are this, like they go into nature, they observe nature, and they
That's fine,
try
but
to.
you're not going to go into nature, right?
Yeah.
That's a job. I'm not saying that that's not something
I read
interesting
a
to
lot
do.
for sure. For anything that I have to write my papers, I read a lot. So if people, okay, I don't go in nature. I know people who goes. They produce the things, and I read. I try to build things that try to explain what they are saying.
I understand, but the question is, will you be able to convince somebody that you've explained it? Because you have to predict something, and it's hard. Of course, it's easy to post-dict. You know, somebody tells you, well, we have these species, I can build a model that explains it. But you're not going to be able to say, no, I predict you're going to see this.
Yeah, well, maybe
you-
I'm looking like into the part. Yeah, I have this dilemma, kind of. That's why maybe I want to build more quantitative to make.
Yeah, that
But
sounds right.
if I say to you, like, I work through curiosity, I think that this is
Sure.
very interesting. Like when I start to read, for example, for the next work that I want to do, the relation between mitochondria and evolution of sex and evolution of eukaryotes is not something that someone can test. But that would be very nice to draw a story, okay, I can tell a story to explain how these things are
Well, maybe
connected.
what Bill suggested, I mean, maybe you can test it by doing biochemistry or something and not.
Yeah, I think at some point there is some labs that are able to build the eukaryote in the lab, but again,
Okay.
we are like this happened in nature, like for a lot of.
Okay.
It's not life. It was not like a lightning, and you put there, and life comes out. Like
So you think in
there's a
5
lot of error when eukaryote appeared. Like
Are you thinking in 5 or 10 years that you're going to be working on this? Is that what you think? You don't know, of course, but.
yeah. I don't. I think I'm a curious person, and in the past 3 years, when I've been studying other things, because one of the things that I wanted to start these conflicts, I wanted to do the more quantitative work, so I started to be interested in conflicts, more general speaking, so I started to work with evolutionary game theory, so I want to also maybe using these tools to apply for other things. So, of course, I want to diversify, because I get that maybe at some point, this is exhausted. But one thing that I enjoy in this area is, when I mean like complex systems, it's nice that you feel like when you learn some tool, you can apply in different contexts. So, for example, when I start to work with this model, these chains, and then that's when I saw this paper from the guy who I work with in US, Josh. He used the same kind of model to explain how antibodies and virus can develop virus can escape the action of antibodies. And this is the same kind of model, zeros and ones, and you have the whole calculation. So I think that this is interesting. You can use like the same tools for different things.
Okay.
This is one that I like, but I get you. This is my work, so I have to something that look useful.
Okay. Thanks. Other questions? Okay, so thanks, Stefan.
Okay.